In less than a day spent in hospital, this patient's eyesight will be restored. Stem cells from the patient's own eye have been harvested on this simple contact lens. This will be placed on the eye to overcome the damage caused by blinding corneal disease. Within weeks, sight will dramatically improve. It's a world-first technique pioneered by researchers at the University of New South Wales and colleagues at Sydney's Prince of Wales Hospital. The novel procedure, which is simple and inexpensive, offers hope to people with a range of blinding conditions that involve corneal damage from infections, burns, and chemotherapy.
It's a simple technique. It requires a small biopsy from a patient. It requires 5 mils of sera. The cells are cultured over a 10-day period on a contact lens, so it requires no foreign human material or animal products. During the culture procedure, the tissue is firmly adherent to the contact lens surface. The pink solution is like fertilizer for the cells. The cells seem to transfer from the contact lens surface onto the patient's ocular surface.
Once the stem cells have multiplied, the lens is dispatched to surgeon Dr. Stephanie Watson of Sydney's Prince of Wales Hospital.
The procedure is relatively non-invasive. For the patient, they merely come into the hospital for a couple of hours and then they're able to go home afterwards. At the beginning of the procedure, we remove all the damaged cells from the front of the eye. Following this, we take the contact lens with the cells that's been cultured in the lab and simply place it on the front of the eye. Afterwards, the patient uses eye drops and then returns the next day and we examine the contact lens to make sure it's in place.
The researchers are hoping the procedure can be adapted for use in other parts of the eye and even in other organs.
We're very excited about this technique because we think it might be applicable to other major organs of the human body, such as the skin, because after all, the skin behaves in a very similar manner to the cornea.